All right, everybody say hi. You can do better than that. Oh, we have a question.
For the serine, is the serine the entire protein, or is it the...
Serine's an amino acid.
Okay.
All right, very good. We have this diagram. We could actually use for this to pick up. I think this was microbiology. So, what we're talking about here is how we can carry out biochemical reactions, and we're starting out first with catabolic type reactions, things that are, as we say in the biz, spontaneous reactions. And spontaneous, of course, doesn't mean it's happening spontaneously. It just means thermodynamically that's the direction you should put your money on if you're going to make a bet as to which way it's going to go. So from a spontaneous type reaction, what we're doing then is lowering the energy of activation and we'll talk about the mechanism behind that a little bit later, I think. Well, maybe we already did. Regardless, we can lower the energy of activation and make it so that the available energy in the system, in our case, running at 37 degrees centigrade, 100 degrees or 98.6 degrees Fahrenheit, is sufficient energy to make the reaction go. So you don't need a match, you just need to have temperatures at the body temperature. And truthfully, enzymes will work even at colder temperatures, just not as fast, but they're able to lower the energy of activation sufficiently to get the reaction to go forward. All right. Then we start talking about non-spontaneous reactions, the way we can carry out anabolic metabolism and build stuff up. We have to have a slightly different kind of model. There's an energy of activation, but it's actually way higher now. And what we're trying to do is actually carry out some organization. We're starting to build stuff. And one model for that is having an enzyme situated like this, where we have a set of negatively charged amino acids. So what I've drawn here crudely with my little, I don't know what you call this, little figure here, is a protein structure. This is like a three-dimensional protein structure. So we got the tertiary structure, secondary structure, and that's dictated, of course, by the primary structure. And we've got these two spots where the two parts can bind. And what we need to do is slam them together close enough that we can form a covalent bond. We have to put energy into that to stick them in there. And the way we do that is often have a neutral amino acid here that, in the absence of phosphate, doesn't carry much of a charge. But we add a phosphate to it, and all of a sudden, it's got this really big negative charge that's pushing everything around it. And this is a somewhat simplified model, but it works as a pretty good kind of analogy and a generalized way of thinking about it. And so the role of phosphates to impart this negative charge is what drives this this shape change. And so we can have really big shape changes occur. And as we start talking later in the semester about muscle contractions or all kinds of different things, there are all kinds of protein shape changes that are being triggered by this phosphate group. And in the case when I've drawn it here, we have a kinase reaction in which we're actually covalently attaching a phosphate to this neutral amino acid, the serine. And that is going to cause this thing to change shape. The phosphate can then be immediately popped off, and the protein can come back into its ground state, to its relaxed state. And so it just cycles between these two different states. And so that's how we can envision anabolic type reactions occurring, is by incorporating the phosphate from ATP. And as we were mentioning, in this case, we actually have a kinase reaction occurring in which we are covalently binding a phosphate to the serine, and then a phosphatase will remove it. And in other systems, we have a little pocket where ATP can nestle in there. The phosphate can break apart, and the phosphate by itself is imparting that really strong negative charge and causing the shape change to occur. And then the whole thing kind of, the ADP, the base with the adenosine and the sugar, that falls out, and then the phosphate falls out. And then, we're going to have a whole set of other systems in which we can rebuild and add that phosphate back to the ADP to make ATP. So that's the way we drive energy requiring reactions. There are other ways, but this is a really good way to think about it. Did that answer your question?
Yeah.
Cool. Yeah. So, what I'm doing is throwing in a couple of abbreviations in that we were mentioning this is a serine, and you just have to know this is a neutral or at least weakly charged amino acid. The R group doesn't have much of a charge in it. Remember, we talked about the R groups that can be subdivided into acidic or basic. They can be positively charged, negatively charged, polar, nonpolar, stuff like that. And it was just sort of a nerd point that when we get into a little bit more cell signaling and stuff, there is another set of amino acids. The tyrosine is another one that is a R group on an amino acid that is subject to phosphorylation in cell signaling. In addition to the serine, there's also the amino acid threonine, which is another neutral one. And you certainly don't need to know anything beyond basic amino acid structure for our class, but it's probably useful to at least read through the names of the amino acids. First off, they make very good cat names, but that way you're familiar with, if someone says, this is a phenylalanine or an aspartic acid or a lysine, you go, oh, those are amino acids. Okay. And we're actually going to have a decoding exercise, which we take a piece of DNA sequence and translate it into amino acids, and you'll have a whole chart there, so you'll get familiar with that later. All right, so that's how we can invoke phosphates to drive anabolic reactions. And that's a good thing. All right, so one of the things we're going to be doing in lab, probably next week we're going to start doing some stuff, but this lab that we're going to do requires a little bit more pipetting skills, so we're going to actually do a little bit of pipetting practice, and then we're going to get into some enzyme kinetics and some enzyme assays. And there is some very specific enzyme models and enzyme stories that we need to throw in here. So, one of the things that we can do is look at the rate of a chemical reaction and incorporate our discussion of enzymes here. So, this is a classic kind of plot of reaction rate. So, on the y-axis, we've got the reaction rate, and then on the x-axis, we're going to put substrate concentration. So if I haven't defined it, here we are. We got the enzyme, which we know is a biochemical catalyst, typically a protein catalyst, and enzymes act on a substrate. That's the stuff that they're chewing on. And then what they generate is what's referred to as the product. So it ain't like, real fancy terminology. But when we start talking about substrate interaction, we're talking about the thing that the enzyme is chewing on. All right. So we're going to actually be looking at a protease and its ability to break apart a peptide bond. Proteases are real common. Snake venoms are often proteases. And this is what we see. So the rate is going to be something like, molecules of a product per minute, whatever. When we're going to be playing with this in the lab, we've got an assay in which we measure an absorbance change, and so it ends up being absorbance units per minute. And so let's just think of this as product generated per second. I decided to change the times. So that would be a rate. And just as a little note, when you're doing and seeing these kinds of rate equations, you have to realize that the way you establish a rate is by doing a series of experiments in which you look at the product generated, and you do this over a experimental assay time, say like 10 minutes. When you do this, what you're going to see is something that might look like this. That over time, you start making more and more product. Okay. So that would be a single experimental point in determining a rate. And what you then do in order to determine rate is you need to get the slope of this line, that is the product generated divided by the time. And that would be product amount per minute, per second. When you plot that against increasing substrate concentration, this is what we see. It is a canonical, that is, pretty much consistently you're going to see this kind of thing, type of curve when we're looking at an enzyme catalyzed reaction. And I will just tell you that when you're looking at rate graphs, when you see this kind of thing arcing over and then flattening out, that is a key that there is a protein catalytic event going on here. Some kind of protein substrate interaction. And we'll see why in just a moment. Now, just as an alternative, if you're looking at, say, a straight chemical reaction, like how many of us have ever made a baking soda and vinegar volcano? I will just tell you, if you haven't done that, you should. Go into your roommate's bedroom with a big box of baking soda and a jar or a bottle of vinegar, and pour. Actually, probably the better way to do it is to go to your kitchen, take a soda bottle, pour a bunch of baking soda, about that much in there, then go into your roommate's room, set it up on near his bed, and pour vinegar into this. And of course, what happens is vinegar is going to cause an acid-base reaction with the baking soda. Baking soda is a base, a weak base, but it decomposes to generate carbon dioxide, and you get this big foaming mess. And all right, so what you're doing is generating carbon dioxide by reacting it with the acid. This is a standard acid-base reaction. And if you were to measure the amount of carbon dioxide, and what you're going to do is have, a liter of vinegar, and you put in a little pinch of carbon dioxide, of baking soda, you see a small amount of CO2 coming out. You add some more, you add some more, you add some more. You just keep on doing this experiment over and over and over again, adding more and more baking soda. What you would see is something that would look like this. So a linear as opposed to a logarithmic. A linear equation is going to be an indication of a non-enzyme catalyzed chemical reaction. You're just talking about a plain old chemical reaction. Another way of looking at this would be if you know that, just as an example, ATP spontaneously degrades and generates phosphate. As I was mentioning in the video, that ATP is one of these molecules that is really delicate, that we have to keep it in the minus 70. You can't freeze thaw it much. I mean, if you look at it funny, it falls apart. And if you take 1 gram of phosphate, of ATP, and after 5 minutes or whatever your assay is, you measure the amount of phosphate released, you'll have this amount. Now, if you take 10 grams, you'll have 10 times that. That would be a non-enzyme catalyzed reaction. And so non-enzyme catalyzed reactions are simply dependent on the amount of stuff, the amount of substrate you got. All right. In an enzyme catalyzed reaction, we have this really cool rate where it starts out really fast and then it arcs over. And what we have is a couple of terms that fall from this. First, this is referred to as the maximum velocity. And in the parlance, this is known as V max. So, a couple of things as well to keep in mind here. This is an enzyme catalyzed reaction. Every single multiple point on this curve is a single assay. And you get the slope of the line, and then you're able to plot this curve. So you don't get this in a single experiment. You got to do a bunch of assays. Additionally, you have to make sure that when you're doing these assays, you're keeping the enzyme concentration constant all the time. That never changes. What you're changing is the substrate concentration. As you increase the substrate concentration, you keep the enzyme the same, and you see this standard reaction. That's how you know it's an enzyme catalyzed reaction. All right, so one of the things that then follows from this is if you take the substrate concentration at 1/2 Vmax, and you might say, why? Well, that's just how it is. In that, it is just a standard point that is established, 1/2 Vmax, and then you look at the substrate concentration. This is a special value that is known as the Michaelis constant, and is named after two women enzymologists back in the 30s, probably. Michaelis constant. So it's Michaelis and Minton. I don't want to give Minton, I don't want to shortchange her. The Michaelis constant is abbreviated K sub m, and that is equivalent to the substrate concentration at 1/2 Vmax. So 1/2 Vmax is going to get you kind of in the steep part of this curve. That's the whole reason we do it there. All right. We'll talk about the significance of Km as we continue on, but these are some terms that we want to throw in. In a general sense, Km is a cool sort of relative binding affinity. Things that have a very high Km are things that don't bind real well. So you can have, an enzyme that doesn't bind to its substrate very well, or you can have a substrate analog, like a drug, that doesn't bind to the enzyme very well. And when you carry out these assays, the Km for that substrate is going to be a specific number, and depending on how well it binds, it may be higher or lower. And then, of course, in the cell, you've got the naturally occurring substrates and whatever they're doing. So the way we think about this then is sort of as a general truism, a lower Km equals a higher binding affinity. And this again is kind of a cool idea that different protein or protein substrate interactions can have different Kms. Stuff that has a really high binding affinity, that is, got a really low Km, it's going to interact really tightly and really fast. Stuff that's got a really low or really high Km and low binding affinity, they don't bind that well. They might knock into each other but not grab hold. The binding may be really loose. Different things can happen. And so Km is a relative binding affinity. And as we've been talking about this, all of this biology we're going to be getting into is all about the way proteins and other molecules interact with each other. Some things have a real high affinity, real specific tight binding. That would give you a low Km. Other things have a fairly loose binding, and they're not very specific. They'll bind something that kind of looks like their substrate. And this becomes also a big deal in the pharmacology and the pharmaceutical industry. Lots of different drugs ends up being things that will bind to specific enzymes you're trying to shut down, which actually brings us to the next point of the enzyme inhibitors. So, let's see. People will take statin drugs to lower their cholesterol. These are drugs that will bind to an enzyme that's involved in cholesterol metabolism. And they'll slow that enzyme down and make it so you make less cholesterol. And not all drugs are interacting as enzyme inhibitors, but pretty much all drugs are interacting with something, like your various pain relievers. They are either inhibiting the production of inflammatory signals, that's one of the things Tylenol does, or they are blocking the receptor for those signals, that's one of the things that aspirin and ibuprofen does. And you can have all kinds of pharmacological interactions by either inhibiting a hormone or blocking a receptor from binding the hormone or slowing down an enzyme or speeding up an enzyme. And so there's lots of different things. And so drug makers are constantly looking at molecules and their Km with different kinds of enzymes and things. All right, so we got this set up now. And from here, we can throw in a couple of sort of classic terms and see how they fit in. So, no, that was a squeak, not a question. You squeaked. Okay. That was a squeak, everybody. Of course, what the Monday and Friday people know is there's a huge amount of communication that I do off screen, and I write out all the test questions over here when I was doing this. And then I come back on, and they've all been sworn to secrecy. And so, I'm sorry, but I like them better. No, that's not true. Okay, so let's talk a little bit about enzyme inhibitors. And we can classify enzyme inhibitors in a couple of different ways. Now, some of this is just ways ways of monkeying around with enzymes, but then we also have relations that we can throw in to the way metabolic pathways are being controlled within your body. So it's worth talking about. Enzyme inhibitors. As we're going to see, at least from our model, we can talk about two different types. The first is called a competitive enzyme inhibitor. Competitive. And then the second is a non-competitive. And we got a couple of ways of defining these fairly nicely. So we can have an enzyme inhibitor. This is going to be something that's going to jack with the enzyme's ability to carry out its chemical reaction. A competitive inhibitor is one that, in terms of poisons that I might take, or that if I had to choose a poison, because lots of poisons end up being inhibitors of enzymes, I always want to choose a competitive inhibitor. And I encourage you to choose competitive inhibitors if you're ever given a poison choice for the following reason. A competitive inhibitor is one that's going to inhibit the enzyme reaction, but we will eventually make our way to the maximum velocity. By adding a whole slug of substrate, we can actually get around the inhibitor and eventually get to Vmax. So we define a competitive inhibitor in this way. The maximum velocity, Vmax, is unchanged. But when we look at the equation, since maximum velocity is not changed, 1/2 Vmax is not changed. And beep, 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 beep. We come out here. And lo and behold, the substrate concentration at 1/2 Vmax, which remember that's Km, is a lot bigger. So Vmax is unchanged, but Km is affected. And I would say Km always gets bigger, but I'm scared that if I said that, that's not right. It's got to be. Although I can imagine that. Maybe it's not. Anyway, I think most of the time it probably is. Regardless, what we do know is Km is going to change. The maximum velocity don't change. Now, a non-competitive inhibitor works like this. All right, here's the reason why. If you ask me what poison do I want a competitive inhibitor, because you give me the poison, and all I got to do is saturate myself with some kind of substrate, and I can get around that poison. I can work around it. And so you might have a competitive inhibitor that blocks your ability to take up oxygen. Okay, that's bad. But if you put me on high flow oxygen mask, I can get around it. A non-competitive inhibitor, I don't want that poison. Because a non-competitive inhibitor does this. A non-competitive inhibitor is one that Vmax is never achieved. So, a non-competitive inhibitor, let's see what markers I got. All right. In a non-competitive inhibitor, Vmax is unachievable. Unachievable. Not gettable. And what this, though, often turns out is that because we've got Vmax right here, Vmax has changed, Vmax has dropped, 1/2 Vmax also drops, and we'll see that Km has not changed. So Km don't change. Now, when you get into biochemistry, or if you go get your graduate work in enzymology, yeah, this is somewhat simplified, but in my simple world, this whole thing makes good sense, and so I'm sticking with it. Yes, sir.
Both the competitive and non-competitive, those aren't linear, right?
Those are what?
Aren't linear.
Yeah, neither one of them. I mean, these are still enzyme catalyzed reactions. So we got, let's see. Yeah, this one is not linear at all. It is actually flattening out. The green line is. Here, I'll make the figures a bit rather than dashed lines. One of the things I'll tell you, whenever I'm drawing this, and I'm learning some tricks to make it work, is that you want to have the lines kind of line up the way the story goes, and so you're drawing it by hand, you can't always know that's going to happen. So here is the competitive inhibitor. So the point being, it starts out a lot slower, it goes way slower, but eventually it'll get to the rate that the other guy, the non-inhibited system, will hit. And we'll do the same for the competitive inhibitor.
Wait, so which is non-competitive and which is competitive?
So green is non-competitive, black is non-competitive. My black's not all that great. Let's see if this does better. Yeah, a little bit better. So we can tell which one is competitive. Competitive inhibitor, we hit Vmax. Non-competitive inhibitor, you don't hit Vmax. And then from that, because competitive inhibitor, you hit Vmax, Vmax ain't changed, 1/2 Vmax isn't changed. The non-competitive inhibitor, though, I'm looking at too many different lines. The non-competitive inhibitor is such that because Vmax has changed, 1/2 Vmax by definition changes. I kind of had this line in the wrong spot, but what the hell is that line? Oh, okay, let's do it this way. This is the Vmax for the non-competitive. Okay. So, here is how we can begin to think about the way enzymes work and how these inhibitors operate. Let's say with a competitive inhibitor, say you take an overdose of a drug that acts as a competitive inhibitor, all we got to do is flood you with substrate and you can get around it. If you take a poison or a drug that is a non-competitive inhibitor, sorry, Chucko, you're just going to have to ride it out until your body clears it, and good luck. Like just as an example here, and this is not quite inhibitors, but it does give you a sense about competition in competitive systems. If you ingest methanol, the enzyme alcohol dehydrogenase is going to turn methanol into the 1-carbon aldehyde formaldehyde, embalming fluid. And apparently your eyeballs are really sensitive to formaldehyde, and it is, in fact, becomes the leading cause of induced blindness. That's why you don't drink methanol. The enzyme can be flooded with 2-carbon alcohol, ethanol, to generate the 2-carbon aldehyde acetaldehyde. Now, if you ingest methanol, first, you probably need to get to the emergency room right away. Second, what they do is they add a competitor to this enzyme system in the form of ethanol. And they flood the system with that, and this poison now is not going to be, it's going to be flushed out of your body, by your kidneys and liver, fast enough that it doesn't get, incorporated into formaldehyde. And so what they do for methanol poisoning is they get you as intoxicated as fast as they possibly can, like, IV vodka. So that's one of the things you can sort of think about in terms of competitive versus non-competitive systems. Let's see. All right. So that's what we got here. And now a little analogy time, and we can think a little bit about how these systems might work. So one of the critical points behind these assays is the way you set the experiment up such that every point on these lines, you're using the same amount of enzyme. If you change the enzyme concentration up, you squirrel the experiment up and it won't work. And what we can think about then is how competitive versus non-competitive inhibitors are interacting with the enzyme. And this is not the only way, but it is a good analogy way. Let us imagine that we as a classroom of 1, 2, 3, 4, 5, 6, 7, and I'll contribute 8 peoples, we are Twinkie eating machines. We eat Twinkies as fast as we can. And we're going to measure the rate of Twinkie eating by looking at the number of wrappers we throw out the window. All right. So we're going to have our first experiment. And through that back door, we have our Twinkie chute, and 4 Twinkies come in. We leap on them, kicking each other. The wrappers fly off, go out the window like that. All right. There weren't enough Twinkies for all the enzymes. Then we get 10. Now all of a sudden, wham, we're all on them Twinkies, and somebody gets 2. The wrappers go out the window at a really fast rate, pretty much almost as fast as they did at the very beginning. Now we're going to start stepping up the substrate concentration. We're getting further out here. Instead of 10 Twinkies, let's go ahead and put in 50 Twinkies. Woo! We start eating Twinkies, man. You get to where you got a Twinkie in one hand, ripping the wrapper off, shoving it in, ripping the wrapper, and you start shoving those wrappers out the window. But at that point, we have saturated the system. And you bring in 50 Twinkies, we're eating them as fast as we can, but man, they ain't no more mouths. You put in 100 Twinkies, well, hell, I can't eat Twinkies any faster than what I'm eating. We have reached the maximum velocity. So I'm eating them as fast as I can, put in 200. I'm still eating them as fast as I can. And so the line flattens out. You've reached maximum velocity. Okay, We then can carry out all the different calculations. We can look at what the maximum velocity is, which we'll say is 10 wrappers per second. We're eating a lot of Twinkies, so we're eating them really fast. And we can then establish a KM for these Twinkies. Now, if you were to take, like Little Debbie's, or and they used to make ding dongs in foil wrappers, different kind of wrapper. We can still eat it, but the wrapper is a little bit different. Our KM and our rate might change. Sure, whatever. Now, let us imagine that we throw in 10 Twinkies, but one of those Twinkies is wrapped in Tyvek house wrap. It's not the plastic cellophane. It is like heavy, heavy plastic, and it is sealed up. And so whoever grabs that one is like, shit. They're yanking on this thing, and they can't get their Twinkie out to dump the wrapper out very fast. They're tied up. Now imagine if we have, 2 or 3 of us jacking with these Twinkies, that's going to be a problem. So like, half of the Twinkies that came in are Tyvek wrapped. It's going to slow us up. But let's just say, for example, that we now have that many Twinkies that are Tyvek wrapped, but when you put in, the next truckload, 100, you grab one and you go, oh, to hell with this, and you grab another one and you rip it apart. And so if you put enough good Twinkies in, I can overcome those funky Twinkies. That would be a competitive reaction. Now, a non-competitive reaction would be kind of a hotshot Twinkie. We got Twinkies coming in, and out of the 10, one of them is laced with, either a sleeping pill or poison, whatever. And all of a sudden, one of our Twinkie eaters, or maybe even, how about this? It is, rather than being a happy Twinkie that you can chew, it's made of foam rubber. And the rule is you can't throw the wrapper out until you swallowed that Twinkie. So you're sitting there gnawing on that damn Twinkie. You can't chew it. And so after a while, we got a bunch of us chewing on rubber Twinkies and we're stuck. Even better would be if they're poison. You eat one and you go, blam, and you fall out. And we look around the room and go, damn, that guy had a bad Twinkie. And so we start getting more in, but you're dead. And so even though we bring more in, we can't increase the rate and get it up to our maximum rate because we actually have taken our enzymes out of the game. And that is how we can think of a non-competitive system. So a non-competitive enzyme inhibitor system is going to be one that actually is taking the enzyme and doing something to it to make it so it can't operate very well. Another way of saying this is a non-competitive enzyme is probably going to affect the active site in the enzyme. That is, the mouth don't work so good no more. A competitive enzyme is probably something that is going to be an altered substrate, but has no effect on the enzyme. So you can have something that you just don't like as well. The spinach flavored Twinkies, or a different wrapper type, whatever. And it's not affecting the enzyme per se. It is simply affecting the enzyme's ability to break it down as fast. In fact, as I think about this, the spinach flavored Twinkie is a perfect example. Because you eat that, and it's just like, boom, oh, that is nasty. And you guys stop and take a drink of water, and you're like scared to eat another Twinkie because it's like getting a earwax flavored Berkey Bott's every flavored bean. Do we watch Harry Potter at all anymore? I don't know if you saw that, but that's one of the prerequisites for the class. You need to have watched all of the original series Star Trek. You need to be familiar with the movie Avatar. You need to be reasonably familiar with The Lord of the Rings, and you need to be reasonably familiar with all the Harry Potter stories.
No Star Wars?
Right.
I'm sorry. That's for genetics. Wow. I could try and incorporate it, but no, I don't think that's not a prerequisite. I've never seen that as a prerequisite before. Star Wars, goodness. Knowing your Marvel Cinematic Universe could be helpful, but that's that's like if you've already had second semester organic chemistry, you're a little bit ahead.
Yeah.
Same thing.
Yeah.
No Star Wars. I'm totally down with the original 6. The new 3. I have to admit, I haven't even watched the last one. But I've been forced to watch all of Clone Wars, all of the Star Wars animated, whatever the next one is, and now we're into this 3rd one, and yeah. But I digress. All right, so now, this is enzyme kinetics, in which we're not jacking with the enzyme too much. We're throwing in inhibitors, yes. But we have another thing that we can think about. Km is unchanged. There should be a D there. Thank you. All right. Looking at rate equations, though, it ain't just for Km. Oh, no. It's not just for breakfast anymore. We can actually look at a number of things by looking at rate equations like this. We can determine an enzyme's optimum conditions. Just as an example, let's look at something that might go like this, in which we got rate, and now let's look at temperature. So this is not going to be the same thing as substrate concentration. We're not going to be able to get Km and stuff like this. This is looking at the physical characteristics of the environment in which this enzyme is working. So how well is the air conditioning working in our Twinkie eating machine? And just for the sake of numbers here, we'll put 0 degrees and 100 degrees, and of course we're working in degrees centigrade. Okay, so as we said, pretty much there's no enzymes on the planet can handle 100 degrees because they're being held together by the weak-ass bonds, and hydrogen bonds can't take that. But if we look at the rate, at 0, the enzyme may not be working much, but doing some. We're going to have a rate that'll look like that. And we can actually explain this pretty darn well. Let's go ahead and say what this is. 50, this is 25, whatever. And this is a guy that operates best at room temperature. I like that, actually. If you want to plot it so it's at 37, whatever. This is an enzyme that has its maximum rate at this mid-range temperature. And it's too cold here, it's just right here, and it's too hot here, and then, heck, out here it's probably being denatured. And so when you look at different kinds of conditions, you get rates like this. And so you can do the same thing. You can do temp. If you look at pH, you'll have something. The salt concentration. These are standard kinds of parameters. And we might end up with all kinds of different sorts of rate equations. Sometimes things will have a really narrow range that they work in. Other times, like salt concentration, you might see something that, there's a real wide range in which the enzyme works. What we can begin to do then is, by carrying out these different assays, is we can identify the optimum conditions for a given enzyme. Now, we're not changing the substrate concentration. What we're doing is changing the environment that the enzyme is in. And honestly, what we're doing here is we're jacking with the secondary and tertiary structure. I mean, this is a way of showing that, yeah, if you monkey with the secondary and tertiary structure too much, you won't have a functional enzyme. You will denature the protein. You'll lose secondary and tertiary structure once you get, around, above or below the optimum. And while we'd like to think that all enzymes are going to be optimized for the same conditions, they're not always, but they're pretty close. And so for you and I, most of the enzymes we work with are going to have a pH optimum, probably around 7.2, and they probably have a salt concentration around 0.9% salt, which is kind of your standard physiological saline. They're going to have a temperature optimum, probably somewhere around body temperature. Yeah, so it's not always exactly that way because you can have enzymes that can work somewhat outside their range, but that's what you typically look at. So, Arctic shrimp, you might very well find they've got some enzymes that are actually optimum at colder temperatures. And certainly we start looking at bacteria that are thermophiles. They've got enzymes that don't even work below 50 degrees. They only work, when you get up into the higher temperatures, 72 degrees and things. Was one of the enzymes I work with. All right, so that's another way we can look at enzymes and its conditions and things. And this specifically is getting at secondary and tertiary structure. All right, that's what we had to say for today, and that's all I know. So we're not doing lab this week. You didn't miss anything yesterday, and we'll see what we do next week. Ciao.